Welcome to the deep dive. So this one today is kind of custom designed for the learner.
Hmm.
Like whether you're, prepping for a biology exam, or maybe you're just trying to understand a recent doctor's visit, or you're just insanely curious about how the world works, you are definitely in the right place.
Absolutely.
Have you ever, looked at a medical chart or heard a doctor just rattle off a diagnosis and felt like they were speaking a completely secret language?
Oh, yeah. It creates this real barrier, honestly.
Right. You're just sitting there in the exam room looking at this paperwork, and it just feels like this impenetrable wall of impossible words.
Yeah, medical terminology has this entire architecture that, frankly, can leave a patient feeling entirely on the outside looking in. I mean, it's intimidating.
So today, we're basically handing you the decoder ring to the two most important puzzle pieces in that secret language. We're going on a deep dive into two little roots. There's cyto, spelled C-Y-T-O, and histo, spelled H-I-S-T-O.
And those two roots, they're the vocabulary backbone for literally everything happening in your body just below the level the naked eye can see.
Right.
If you think about, the hierarchy of life, you start with atoms, then molecules, then the organelles inside your cells. Cyto and histo represent those crucial middle rungs on the ladder right before you zoom out to whole organs and, well, full organisms.
Okay, so give us the basic definitions first.
So cyto is the root for cell, and histo is the root for tissue.
Which just sounds incredibly clean and orderly when you read it in a textbook. Like, a cell is the smallest functional unit of life, and a tissue is a group of those cells working together.
Sure, that's the textbook version.
But here is where it gets really fascinating. Those neat, tidy definitions that we just take for granted today, they're actually the result of centuries of bitter scientific rivalries, grave-robbing anatomists, and a few truly mind-blowing happy accidents.
Oh, the origin story of these words is wild. Because to understand how a modern oncologist uses these terms today to diagnose something like cancer, we kind of have to look at how the words were coined in the first place.
Right.
Both of these roots actually started out meaning something entirely different from what they mean today.
So let's start with cyto, the cell. Where does this word actually come from?
Okay, so the Greek root is kaitos. But in ancient Greece, kaitos did not mean the basic unit of life.
Wait, what did it mean then?
It meant a hollow vessel, or like a container. It was purely a description of a physical shape, usually something empty inside, like a jar.
Okay, wait, if it just means an empty jar, how on earth did it become the defining word for the complex building blocks of all living things? It's quite a leap.
Well, for that, we have to travel back to 1665 to an English scientist named Robert Hooke.
Okay.
So Hooke was playing around with a very early, very rudimentary compound microscope, and he decided to put a very thin slice of cork under the lens, and he published his observations in a famous book called Micrographia.
Oh yeah, I remember reading about micrographia.
Right. So under the magnification, he saw this rigid lattice of small, box-like compartments, and he called them cells because the repeating geometric shapes, they reminded him of the small, bare, unadorned rooms that monks lived in at monasteries.
Oh, like the monk's cells.
Exactly.
Yeah.
Which were called cellulae in Latin.
See, this is such a brilliant historical irony to me, like biology's great origin story. The man who discovered the cell, the guy who gave it the name that every high school biology student has to memorize today,
Yep.
he never actually saw a living one.
No, he didn't. He was looking at dead wood. Cork has no living biological material inside of it at all.
Wow.
When Hooke looked through that lens, all the cytoplasm, the nucleus, the actual living machinery of the plant had long since dried up and vanished. He was literally just staring at the dead, empty cellulose walls left behind.
So he was looking at the shape of an empty container.
Precisely, which ironically fit that ancient Greek root, kaitos, perfectly. It took over 170 years for the scientific community to actually evolve the word cell from a description of an empty shape into this profound theory of living matter.
Okay, so Hooke is looking at empty rooms. What about histo, the root for tissue?
So the Greek root there is histos, which translates to web, or that which is set upright.
Web.
Yeah, in its original context, it actually referred to the upright warp threads on a weaving loom.
A woven fabric. Okay, that actually makes visual sense. Like, if you pull apart muscle or connective material, it does look fibrous and woven together.
Exactly. So it entered early medicine as a metaphor for that fibrous appearance. But the person who truly transformed the concept of tissue into a foundational medical science was a French anatomist named Marie-François Xavier Bichat.
Okay.
He was working in Paris in the 1790s, and honestly, his sheer output just defies belief.
How so?
Well, Bichat hypothesized that whole organs were not the fundamental unit of the body. which is what everyone previously thought. He believed organs were built out of simpler, distinct building blocks, which he called tissues.
Okay, so how did he prove it?
To prove it, he dissected an extraordinary number of human cadavers. By his own meticulous account, he dissected over 600 cadavers in a single winter.
Sorry, but 600 bodies in one winter?
Yeah.
The sheer logistics of acquiring and examining that many cadavers in 1790s Paris is terrifying.
He was a man possessed, truly. And his methodology was intensely physical. I mean, he didn't just slice them open. He tested the structural limits of the body's materials.
What do you mean by tested?
He would literally boil slices of organs in water or, submerge them in strong acids or apply direct heat or let them deliberately putrefy.
Oh, God.
Yeah, and he'd observe whether the material dissolved into a jelly or hardened into a tough fiber or shriveled up. And by analyzing these chemical and physical reactions, he identified 21 distinct fundamental tissue types.
Like what?
Like membranes, cellular tissue, glandular tissue, and so on. He published this masterwork in 1800.
But wait, I have a question about the timeline here. If Hooke is using a microscope in 1665 and Bichat is working a century and a half later, how is Bichat identifying 21 microscopic structures?
Well, he wasn't. Bichat flatly refused to use a microscope.
Seriously.
Yep, he deliberately banned them from his laboratory.
Why? I mean, wouldn't a microscope be the ultimate tool for someone trying to find the smallest components of an organ?
You'd think so, but you have to remember the technology of the time. Late 18th century microscopes suffered from severe chromatic aberration.
Oh, right, the lenses were bad.
Yeah, they produced halos of false color and super blurry edges. Bichat thought they were methodologically unreliable. He argued that when you look through a distorted lens, you aren't seeing biology, you're just seeing instrument artifacts.
That makes a lot of sense, actually.
Right. He insisted that true science required the naked eye, the hands, and chemical reactions. But, tragically, spending his days and nights surrounded by hundreds of putrefying bodies took its toll.
Oh, no.
He died in 1802 at the age of 30, likely from a severe infection he contracted during a dissection.
Wow. He literally gave his life to this idea, mapping the human body by hand.
He did, and he died before cell theory even existed. But the linguistic twist that happened a few years later is remarkable.
What happened?
Well, in 1819, a German anatomist named Karl Mayer needed a new word to describe the microscopic study of the body's structural materials. So he took the Greek root histos and coined the word histology.
Let me guess.
Yep, he specifically created the word to name the microscopic study that Bichat, the founder of the field, had stubbornly refused to do.
The entire scientific field of histology is named after the exact methodology its founder hated most. That is phenomenal.
It's pretty funny, yeah.
But it leaves us with a huge missing link, right? We have Hooke looking at dead shapes through a microscope, and we have Bichat categorizing macroscopic webs by hand without one.
Mm-hmm.
How did anyone figure out that Bichat's macroscopic tissues are actually made of living versions of Hooke's empty cells?
That synthesis required the biological smackdown of the 19th century, which was the birth of cell theory. In 1838, a German botanist named Matthias Schleiden finally proposed that all plant tissue is composed of living cells.
Okay.
And then 1 year later, his colleague, a physiologist named Theodor Schwann, extended that groundbreaking claim to animals. Together, they established the paradigm that the cell is the universal, basic, structural, and functional unit of all living organisms.
But even if they realize that everything is built of cells, where do they think those cells came from? Because, like, at this point in history, spontaneous generation was still a massive accepted scientific theory, wasn't it?
Oh, absolutely.
The idea that life could just poof into existence from non-living muck.
It was very popular. Even some of Schwann's smartest contemporaries believed new cells could spontaneously crystallize out of a non-living biological fluid they called blastema.
Blastema. Wow.
Yeah, and it took another couple of decades to kill that idea. In 1855, a brilliant German pathologist named Rudolf Virchow dropped what might be the ultimate mic drop in the history of biology.
What did you do?
He published a Latin dictum, omnis cellula e cellula. Every cell arises from a pre-existing cell. And he later cemented this in his 1858 landmark textbook, Cellular Pathology.
Okay, so if I'm trying to understand the history of medicine, why is that specific sentence, every cell arises from a pre-existing cell? Why is this such a massive turning point?
Because Virchow wasn't just stating a neat trivia fact about biology, he was fundamentally relocating the origin of human disease.
Relocating it.
Right. Before Virchow, Western medicine largely believed disease was caused by an imbalance of the four humors. Blood, phlegm, black bile, and yellow bile.
Oh, right, the humors.
If you were sick, the humors were out of balance, which is why doctors would bleed patients or induce vomiting. Or, alternatively, they just blamed disease on whole organs failing mysteriously.
But Virchow changes that.
Virchow proved that disease doesn't start in the humors, and it doesn't start at the organ level. It starts when a single individual cell malfunctions.
Wow.
You can't fix an illness by balancing fluids. You have to find the specific cellular tissue that is damaged. This conceptual leap literally birthed modern pathology.
I see the elegance of Virchow's rule. Like, a cell is a complete self-reproducing unit. Every cell comes from a cell.
Exactly.
But if we fast forward to modern science, does that ironclad rule actually hold up? Because biology always seems to have exceptions.
Oh, definitely. The boundaries are genuinely blurry, and the debate over what strictly counts as a cell is surprisingly active today.
Really? Like what?
Well, consider mammalian red blood cells. As they mature, they eject their own nucleus to make more room to carry oxygen, so they have no DNA of their own. Are they complete cells?
Huh. I never thought of that. They can't reproduce themselves if they don't have DNA.
Exactly. Or what about things like skeletal muscle?
What's the exception there?
Skeletal muscle and placental tissue form what is called a syncytium. Instead of individual cells separated by neat membranes, you have a single massive outer membrane enclosing hundreds or thousands of nuclei, all sharing one giant pool of cytoplasm.
That's wild. Is that one single giant cell, or is it many cells that melted together?
Modern histology treats it pragmatically as a multinucleated cell, but it thoroughly challenges that clean textbook definition we talked about earlier. And then, of course, there are viruses.
Which Virchow knew nothing about, because they weren't even discovered until the 1890s.
Right. And viruses completely defy Virchow's rule. They have no cellular structure. They don't have the machinery to generate their own energy or reproduce.
They just hijack things.
They're basically just rogue packets of genetic code. They have to inject themselves into a host cell and hijack the host's machinery to copy themselves. Virologists and cell biologists still fiercely argue over whether viruses should even be classified as alive.
Okay, so we have this rich history, and we have these fascinating blurry boundaries at the edges of the definitions. Let's bring this directly into the doctor's office today.
Let's do it.
Virchow established the cell as the root of disease. How does this actually play out when you go get a checkup? Like, how do cytology and histology function as actual detective tools?
Well, let's start with cytology. In a clinical setting, cytology involves diagnosing disease by looking at isolated individual cells without needing to see the surrounding tissue architecture.
Right.
And the most historically significant example of this is the work of George Papinecolau.
Oh, the inventor of the Pap smear.
You got it. In 1928, and later in a definitive 1943 monograph, Papinecolau proved that you could detect cervical cancer long before a tumor was visible or symptomatic.
Just by looking at the cells.
Yep. His method was to examine individual exfoliated cells. Those are cells that naturally slough off the surface of the cervix under microscope.
But what is he actually looking for? I mean, if he's just looking at a scraped-off cell, how does he know it's cancerous?
He was looking at the internal mechanics, specifically the nucleus. A normal, healthy cervical cell has a tiny nucleus surrounded by a huge, healthy ocean of cytoplasm.
Right.
But Papinecolau noticed that when cancer is brewing, that ratio completely flips. The nucleus becomes massive, irregular, and hyperchromatic.
Hyperchromatic meaning it stains really dark.
Exactly, because it's packed with chaotic, malfunctioning DNA. The cytoplasm shrinks, the whole cell shape distorts. And by identifying those specific abnormal nuclei, he created a screening tool that eventually reduced cervical cancer deaths by roughly 70% in screened populations over the subsequent decades.
70%. That has to be one of the most successful cancer screening tools in the history of medicine.
It absolutely is.
And it is built entirely on the cytoroot, just looking at isolated individual cells.
But cytology is just an early warning system, and it has distinct limits.
How so?
Because if a pap smear flags something suspicious, like if the lab says, hey, these individual cells have enlarged nuclei, the doctor doesn't just immediately start radiation.
Right, you need more info.
The next step is almost always a biopsy. And a biopsy shifts the investigation from cytology over to histopathology.
Ah. So if I'm understanding the difference here, Being a cytologist is kind of like being a building inspector who picks up a single brick lying on the sidewalk. You look closely at it, and you notice the clay is crumbling. You know something is fundamentally wrong with the manufacturing of that specific brick.
Exactly.
But histology, or histopathology, is zooming out to look at the whole brick wall.
That captures the transition perfectly. When a surgeon performs a biopsy, they aren't scraping off loose cells. They remove a small, solid piece of intact tissue.
The wall.
The pathologist puts that slice under the microscope. They already know you have some crumbling bricks. Now they need to check the structural integrity of the wall.
What are they looking for?
They look to see if the normal, orderly, layered architecture of the glandular tissue has broken down into a chaotic mass. And crucially, have those abnormal cells broken through the basement membrane.
The basement membrane being the foundational boundary of the tissue layer.
Right, which is the defining technical difference between carcinoma in situ, which literally translates to cancer sitting safely in its original place, versus invasive cancer that is actively spreading.
That is the literal difference between a localized problem and a massive life-threatening crisis.
It is. Cytology raises the early warning flag, but histology confirms whether the invasion has begun.
It's amazing how those two roots work together like a tag team. So we've talked about how cells and tissues function normally and how we detect when they start to break down.
Right.
But the real power of understanding this medical vocabulary comes alive when we look at the extremes, right? Like when cells break the rules entirely.
Oh, absolutely.
Let's look at the story of Henrietta Lacks.
Yes. Her biology fundamentally transformed modern science. In February 1951, a biopsy of cervical cancer was taken from a woman named Henrietta Lacks at Johns Hopkins Hospital.
And biologically, what happened with that specific tissue sample was completely unprecedented.
Totally. Before her sample, almost every human cell sample cultured in a laboratory would undergo a few divisions and then inevitably die. Human cells have biological clocks.
They can only divide so many times.
Right. But her cancer cells didn't stop. They just kept dividing indefinitely. They were named HeLa cells, using the first two letters of her first and last name, and they became the world's first immortalized human cell line.
When we say immortalized, we mean they have been dividing continuously in laboratories for over 70 years.
70 years.
They were crucial in developing the polio vaccine. They are a cornerstone of global cancer research, and they were used extensively in early COVID-19 research.
It's staggering. Because HeLa cells have been distributed to thousands of labs worldwide and they've been multiplying exponentially for 7 decades, the total mass of HeLa cells ever grown is estimated to be vastly greater than the weight of Henrietta Lacks's own body when she was alive.
Let's just pause and imagine that. A single tumor's cyto-level abnormality has outlived and completely outweighed the human being it came from.
It's incredible. And this connects beautifully back to Virchow's big realization.
Oh, so?
Well, in the 19th century, scientists debated whether cancer tumors were foreign parasites invading the body or something else. Virchow's cellular pathology framework helped us realize that cancer is a disease of a cell's own internal genome malfunctioning.
Oh.
It's an internal mutation causing unchecked growth, not an external contaminant. That conceptual shift underlies every single modern targeted cancer therapy we have.
Wow.
And once you grasp cyto and histo, the whole medical dictionary really just starts to unlock for you. If you bolt a suffix onto these roots, suddenly you can read that complex medical chart.
It's like Lego blocks. Like if you add ebplasm, which means formative substance, you get cytoplasm, which is the fluid matrix inside the cell where all the organelles float.
Exactly. And when a cell physically splits in two during reproduction, that physical movement is cytokinesis, kinesis for movement.
Right.
When a cell ruptures and breaks apart, it's cytolysis, meaning breakdown.
And if you add shadegenesis for origin or creation, you get histogenesis, the complex embryological process of how different tissue layers actually form in a developing fetus.
You're getting it. And if you combine it with prefixes like kario or nucleo, which refer to the nucleus, you get cytogenetics.
Cytogenetics.
That is the study of chromosomes inside the cell. It was exactly this field that allowed the French geneticist Jérôme Lejeune to identify trisomy 21, the genetic cause of Down syndrome, simply by counting chromosomes under a microscope in 1959.
You really can build a whole medical universe from these roots. But before we wrap up this deep dive, let's clear up some major misconceptions.
Let's do it.
Because even seasoned biology students hold on to some confusing ideas, and getting this right is key to understanding today's medical headlines.
Oh, for sure.
For instance, when I first learned this, I always just pictured a whale as having gargantuan cells compared to a mouse. Like, Bigger animal, bigger building blocks.
It seems like an intuitive assumption, right? An elephant should have massive beach ball-sized cells compared to a rodent.
Exactly.
But a blue whale's cells are roughly the exact same microscopic size as a mouse's cells.
Wait, really?
Yeah. The whale doesn't have larger units, it just has vastly, vastly more of them.
So the cell size never changes.
It proves that the eukaryotic cell is a biologically fixed size unit, restricted by the physics of how nutrients diffuse across a membrane. It isn't an infinitely scalable balloon.
Okay, that makes sense. What about the root cyto itself? Does it only refer to blood? Because I hear terms like leukocyte for white blood cell or erythrocyte for red blood cell.
It refers to absolutely any cell in the body.
Oh, any cell.
Any cell. A hepatocyte is a liver cell. An osteocyte is a bone cell. A melanocyte is a skin cell producing pigment. Blood just happens to be the fluid tissue we test most often, so you hear it more.
Ah, okay. When I hear the word tissue, I tend to just think of generic flesh. Fatty tissue, muscle tissue, whatever's under the skin. Is it just a catch-all term?
Scientifically, it is surprisingly restricted. Remember how BiChat categorized 21 different types by hand?
Yeah, with the boiling and the acid?
Well, modern histology has collapsed those down into exactly 4 foundational categories.
Just 4.
Just 4. Epithelial tissue, which covers surfaces, connective tissue, which binds things together, muscle tissue, which contracts, and nervous tissue, which conducts electrical signals.
Wow.
Every single organ in your body is just an intricate combination of those 4 basic structural materials.
And understanding those structural materials is the entire foundation of the most cutting-edge medicine today. Look at CAR-AT cell therapy, which was first FDA approved in 2017.
CAR-T is a masterpiece of applied cytology.
It really is.
They extract a patient's own immune P cells, and they use a viral vector to rewrite the cell's DNA so it grows a new receptor, a chimeric antigen receptor. Then they put those modified cytolevel units back into the patient, where they hunt down the cancer like guided missiles.
Or look at organoids. These are miniature 3D tissue structures grown in a lab to test drugs.
Organoids completely rely on histological principles. Scientists learned that if you give stem cells the right chemical cues and physical scaffolding, they have an innate histological memory.
Meaning what?
They will naturally self-organize into the complex, layered tissue architecture of a miniature brain or kidney.
That is just sci-fi stuff right there.
Yeah.
And of course, we all heard the phrase cytokine storm constantly in the news during the COVID-19 pandemic.
Yep. Cyto for cell plus endocrine for movement. Cytokines are the chemical signaling molecules that immune cells release to communicate with each other.
So a storm is?
A cytokine storm is when that cytolevel communication cascade goes completely out of control, causing massive systemic inflammation.
Man, just look at the incredible journey we just took. We started out trying to decode a confusing medical chart.
We did.
We went from Robert Hooke staring at the empty dead walls of cork that reminded him of a monk's bare room, to a French anatomist boiling and dissolving organs while stubbornly refusing to use a blurry microscope.
To Henrietta Lack's immortal cells outweighing her own body and helping cure polio.
Right up to today's DNA-engineered immunotherapies. By understanding cyto and histo, you aren't just learning vocabulary. You are understanding how your own body is built, brick by brick and wall by wall, and how modern medicine actually goes in and saves it.
It really is a profound shift in how you view yourself. But, I want to leave you with a final thought to ponder, something that pushes the boundaries of everything we've explored today.
Let's hear it.
We talked about how Virchow established the ironclad rule of biology. Omnis cellula is cellula. Every cell comes from a pre-existing cell.
The unbroken chain of life.
Exactly. And we also talked about viruses, which are not cells. They are just rogue genetic code. Yet viruses can slip inside our cellular machinery, hijack our cytoplasm, and fundamentally alter our cellular DNA to the point of causing cancer or driving long-term evolution. So think about this. If viruses aren't technically alive, but they dictate the destiny of the living cells they infect, who is really in the driver's seat of life on Earth? The living cell. Or the non-living code that hacks it?
Oh, man. That is a lingering question that will definitely keep you up at night. Next time you hear a doctor rattling off that secret language, just remember, you've got the decoder ring now. Thank you for joining us on this deep dive. Keep questioning the world around you, and we'll catch you next time.